All right, welcome to the next presentation in our 2019 Corn College series. We would like to welcome Dr. Pankaj Trivedi from CSU, who is in the Biological Sciences and Pest Management Department. He has been part of the research project that we initiated in 2018, basically looking at a pathogen development and some nematode looking at nematode pressure further in some of our continuous corn fields in eastern Colorado. So we're happy to have Dr. Trivedi join us this morning so that he can relay some of the latest results in the study and give us an update. So, Dr. Trivedi, I'll turn it over to you.
Thank you, Chad. So I'll be talking about the development of amplicon sequencing method that we are using to quantify the nematodes and fungi which are associated with crown rot and lodging of corn, and how the different tillage management uh, are impacting the nematode or fungal pathogen pressure. So, yeah. So just to give you a brief introduction, like over the past 10 years, the strip till and other reduced tillage practices have gained popularity in the corn growing regions of Colorado and Nebraska. And these reduced till practices have their own benefits, such as mostly in terms of nutrient retention and moisture retention, which can lead to increased soil health. But they also have some potential negatives, such as the increased incidence of stalk or crown rot in the continuous irrigated corn. And in in Colorado, in the western Colorado and eastern Kansas and Nebraska, we have about 1 million acres of cotton of corn produced, and this is and there's a very less scope of the crop rotation. And this the current management practices in our region appear to have created a perfect environment for the fungal and nematodes to thrive in this situation. And as a result, we have seen that the fields which have little rotation or have been in a strip till system for greater than 7 years, the grain yields have drastically gone down or have become stagnant in the due course of time. And but we there is not much of a research going on in understanding what is the impact of different management practices on the nematodes and the fungal pathogens. And to the best of our knowledge, there has been uh, no work towards the development of a robust diagnostic tool which relies on the next generation sequencing technology and which can be used in the field by the growers. So our objective for our project was to determine the impact of tillage treatments on reducing nematodes and fungal pathogen population and to develop a next generation amplicon-based sequencing diagnostic tool for the identification and quantification of nematodes and fungal pathogens from the soil. So I'll give you a brief methodology, but it's brief because I think Chad, you already have uh, given a broad outline of our experimental plan.
Yes, that's correct. So.
So I'll just go briefly. So our experimental design is we have three experimental sites, which I will refer to GF6, 16, and T1. And in each site, we have three different tillage uh, treatments. We have strip till, which I will refer to ST. We have vertical till plus strip till, which I will refer to VT plus ST. And we have conventional tillage, which I will refer to CT from now onwards. And each tillage strips were replicated 4 times in each field. And this field, what we have identified, have a history of continuous corn production and have been in reduced till system for the past 5 or 10 years. So we collected soil samples in 2018 after the harvest from each plot, and then we brought them into our lab at CSU on ice. And in our lab, we conducted analysis on soil properties, which included the nutrients and soil enzymes. We performed the amplicon sequencing and molecular analysis to understand the fluctuations in the community of nematodes, fungus, and bacteria. And we also performed the morphological identification of plant pathogenic nematodes with Dr. Tim Todd in the Kansas State University. So the first thing is, what in our soil samples we found that the numbers of the plant pathogenic nematode range from 500 to 2000 per 100 centimeter cube of soil. And our site at GF6 has the least number of plant pathogenic nematodes, while the sites at T1 have the highest number of these plant pathogenic nematodes. And interestingly, what we see here is that the reduced till treatments have a significantly higher number of plant pathogenic nematodes as compared to the CT treatment. And let's go ahead and look into what are the different groups of nematodes which are more abundant in different sites and within different treatment among different sites. So we have two major groups in the site GF6. We have five plant pathogenic nematode groups in site T1, and we have seven groups in site 16. And of these groups, two of the more important groups, which belongs to Cunicil, Celsius and Pratalanchus, they were much more abundant in all the three sites, and the relative abundance was higher in the strip till treatments as compared to the conventional tillage. In the site T1, in addition to these two major groups, we have another group called Trichonemoidetes, which was higher in the strip tillage. And in the site 16, we have some additional groups, basically belonging to Hoplolemius, which were higher in the strip till practice. We have an interesting group here is Trichonemoidetes. It was only present in the VT plus ST treatment, and it was not present in any other management in the site 16.
So,
So how
doc,
would the
Dr. Trivedi, if I may quick ask you a question related to just general numbers. It appears like really not much difference overall in the vertical till plus strip till treatment. The strip till, it seems like those two treatments mirrored each other. So most likely we didn't have enough soil disturbance with that vertical till to really make an impact on the nematode numbers. Would you agree with that?
Yes, I totally agree. And then I will show you in the further slide that we found the same trends when we used our amplicon sequencing data, and they those two treatments are, don't have much of a difference except for some few groups. So as you said, like it might be the reason that in the vertical tillage system that we didn't disturb the soil too much, so that the population is still the same.
Okay.
And this is also reflected with the fungal pathogen, that these are also kind of similar for these treatments.
Okay. Thank you.
So as I said, this plant pathogenic nematodes, what we find in our samples, are known to cause damage to the corn fields, such as the stent nematodes belonging to Cunicil, Cunicelsius. These are the generally the mild pathogens, and if their number is above 50, they can cause a significant damage. And that's what we are seeing in some of our samples, that the numbers grows to 50 of the stunt nematodes. We also have the listen nematodes, which were present in all of our soil samples, and they were higher in the tillage treatments, in the VT and VT plus ST treatment, and these are the most important nematodes attacking corn. And one species is reported to be highly pathogenic, but then there are other species within the similar group which has the similar feeding habits and also probably are the good pathogens. In two sites, we found the lens nematodes, which are generally prevalent in the corn growing in sandy soil. And it is being reported that if the number increases to 300 or 400, it can cause damage in the field test. Then we have ring nematodes. Again, this was higher in the ST treatment, and these are very common in sandy soils. We also have Tiger nematode inside 16, which is a very important plant parasitic nematode, and it feeds on the fibrous root, and any number over 30 or 40 is suspected to be causing the disease. And interestingly, we have uh, in our analysis, we found like this sort of numbers present in few of our samples, particularly related to the strip till treatments. In this slide, I'm showing the results from the soil analysis, and uh, what we expected is the soil carbon and the soil nitrogen, or the total organic carbon and organic nitrogen, were higher in the ST and the VT plus ST treatment as compared to conventional tillage. We didn't find any differences in the ammonium concentration in all within the three treatments, and the phosphorus was lower in the VT plus ST treatment in site T1, but it was higher in the site 16 as compared to the other treatment in VT plus ST. And we have to relate this data with uh, sequencing data, and also with our the morphological data to make sense how different species are correlating with the soil properties. So this is the activity of different enzymes related to the carbon cycling and also the phosphorus cycling. And this is we have this data for our two of our site, and more or less the trends are same, that the activity of these enzymes is higher in the strip till treatment, which might be due to the greater amount of carbon uh, and like the great amount of carbon going into the systems, which is utilized by the microbes. So up till now, we have seen the data which is based on the traditional morphological analysis of the soil nematode communities, and this process of traditional this process of like traditional identification of nematode is very labor extensive. It takes a lot of time. We have to separate the nematodes, then we have to look into the microscope, then we have to identify different taxonomic features based on the morphology. So it requires a lot of time, it requires a very high level of expertise, and it is biased towards the dominant groups of nematodes. So to reduce this limitation, we have designed, we have developed a new method based on the next generation sequencing. And for this method, we have optimized the protocol from the MySeq Illumina platform, which is an instrument we use for the sequencing techniques. We have designed some novel primers which can target only the nematodes out of the many different microbes in the soil. And then we have developed our own database, which for the fast taxonomical classification of the nematodes present in the soil samples. So, and this by doing this analysis, we find about 3,000 unique species of nematodes in our soil samples. They belong to 22 different families, as and this is much higher as compared to what we can find out with the traditional morphological technique. So also we compared our data from the sequencing data with the traditional technique, and what we see here is that our new approach was able to identify all the plant pathogenic nematodes what we detected from the traditional techniques, and the relative abundance of these groups was very much correlated what we find with the use of these two different techniques. And then what we did was, now we used our sequencing data to find out how different management practices impact the structure of the nematode communities in soil. So here we see a few principal coordinate analysis, and in this analysis, every symbol is represent one plot, and which is replicated twice, and the red round is the conventional till, the green square is the strip till, and the blue triangle is the strip till plus the VT treatment. And the separation in the communities here represent that the management has a tremendous impact on the structure of the nematode communities in the soil. So what we see is in all three cases, in all three sites, we have a clear separation of the nematode community from the different from the different management practices, which suggests that these management practices have a very strong impact on the nematode communities in the soil. And then we characterize this nematode community based on different functional groups. So what we see here is the fungal feeders and the bacterial feeders were significantly higher in the conventional treatment. And these fungal feeders and the bacterial feeders, they eat the bacteria and fungi, as the name suggests, and they also feed on the pathogenic microorganisms, and by feeding on them, they also release the nitrogen, which can be used by the plants. And the plant parasite, as we have already shown, that these plant parasites uh, are much higher in our strip till and the strip till plus the VT treatments. In the site T1, we have the omnivore or predators, which are the nematodes, which have different feeding habits, have different stages of their lifetime, and these were lower in our strip till treatment as compared to the other two treatments in the site T1. So this was interesting. So this is our data from our uh, the sequencing effort for the fungal communities. And we just have a preliminary analysis of this data. So from the previous study, what we have isolated is different fungal groups from our sites, which are related to the crown rot. And we tried uh, to find the relative abundance of these groups in in different sites and compare them with a different treatment. So we we find that the relative abundance of Fusarium graminearum, Fusarium verticilloides, and Rhizocutina solani, which are associated with the crown rot, were higher in the strip tool treatment. And this is consistent among all the three sites. So what we presume is that there is an interaction between the higher abundance of the plant pathogenic nematodes and the pathogenic fungus, which might play a very big role in the yield in these three different sites.
It's Dr. Trivedi. It's interesting that with the nematode and the fungal data here, it almost seems like that strip till plus vertical till treatment. The vertical till is not very intensive tillage at all, but it's almost like that is although it may not always be significant, it's trending to have a potential even worse impact as far as increase in nematode numbers and pathogens, fungal species here in this case, compared to that strip till only treatment. That's interesting. I would have thought that it would have been the other way around. So interesting.
Yeah, and that's I think I will show it in the next step forward. We have to relate when we analyze the microbial data. We have to relate it with the actual yield and disease incidence in in the field conditions, and that's what we are also proposing, that we have to have multiple time points in which we collect this data. So right now we just have one time point, which was the end of the harvest season. But what we suggest is to also go for another time point in the start of the planting season to look where these trends are similar across the
Yeah.
different points.
Yep. Definitely. Thanks.
So just for to summarize what we have, the data we have till now, that we have, although our study is just a single sampling date, and which was near the end of the corn growing season, but our study helped us to generate a snapshot of the nematode community at a given time point in three sites. We have developed a new method which is very robust and which relies on metabarcoding approach using the next generation sequencing technique that will allow us to look into the soil nematode communities and how these are affected by the management practices in agroecosystems. And by using the combination of our morphological and metabarcoding approach, what we see here is that different management practices have differential impact on both the fungal and the nematode populations in the soil in our soils. And especially we see that plant pathogenic nematodes were significantly higher in the ST and VT plus ST treatments in all of our three sites. And the reduced tillage has an increased abundance of plant pathogenic fungi, which might relate to the higher incidence of the of, of the root of the stalk rot in the plant. And we also acknowledge that we have to build on the foundational knowledge of the nematode communities because there has not been much of a work done to fully profile these nematode communities. And this requires sampling across the multiple time points, as I already said, throughout the season, as well as improvements in the method available for nematode identification. So moving forward, we are still characterizing the bacterial and fungal communities from the soil samples we collected before. Once we are done with this, we will be correlating the microbial community structure with the disease incidence and the soil properties and plant yield in order to find out the indicators of disease incidence or the indicators of soil health. We are planning another round of sampling, and this we propose to do at the time of planting. And after the ultimate goal of the project is to develop a quick diagnostic assay using either qPCR or handheld sequencing device, as you can see in this picture, which the which the growers can just use it in the field and can use a laptop or a mobile device to profile the important microbial members that are related to the disease in our cornfields. And with this, I end my presentation and I acknowledge funding from the Pioneer Seeds.
Thank you, Dr. Trivedi. I think we have very interesting results. I think it's interesting the relatively quick impact that one tillage had on the numbers, and like you said, it'll be interesting moving forward to see how those numbers are affected with another round of tillage. So it'll be interesting to follow that this next year. And then also, yeah, I can't forget to mention the development of a quick diagnostic tool that agronomists or farmers or consultants can quickly obtain a sample from an affected area in a field to quickly identify and quantify not only the number of nematodes or fungal pathogens that may be present, quickly identify that and determine the problem instead of having to wait 1 or 2 or 3 weeks to get that some of that analysis back from the lab. So that's great work. And once again, yeah, I want to thank Pioneer for funding this study and helping us out. And I think with that, we'll go ahead and end the presentation.